Right, okay. So it gives me great pleasure to introduce the next speaker in the Wirral Viking Trail lecture series, and that's Dr. Jennifer Wakefield from the University of Oslo, although you're going to be joining the University of York quite
We'll see, hopefully.
soon. And Jennifer is actually a chemist. She did a degree in chemistry at Aberdeen, and then a master's in analytical chemistry. Yep, and then did another master's in archaeology, Egyptology at Manchester. So she's combined this expertise in chemistry with a great love for archaeology, and then she came to do a PhD with us actually here at Nottingham, between ourselves in biosciences and Rob Stockman in chemistry, in conjunction with the Cultural History Museum at Oslo with Hartmut Kutzke and Susan Barvik on the Saving Oseberg project, designing new consolidants, bio-based consolidants to replace all the rotted cellulose and all sorts, which left not the two or the three main Viking ships under threat, but all the beautiful artifacts, burial artifacts that were found with them under serious threat. So she was involved very much with that and did some great work, and that work is still ongoing. But for the last 3 years, Jennifer, she's been on the what's called the TEXREC project, which is concerning preservation and virtual reconstruction of the textiles tapestries that were discovered with the Oseberg ship. I think over 80 fragments, which belong to several tapestries, of course, in various degrees of disrepair, and all the colour has been lost. But this project has been a wonderful detective story. Well, first of all, try and see how they all piece together, and secondly, to try and conserve them, to reinforce the degraded wool fibre and all this sort of stuff. That the original stuff with some new bio-based materials, rather like the Oseberg wooden artifacts project. But also because these tapestries and things were once coloured, I mean, no colour remains now, apart from a little bit of red, from the degradation products of the original colours, which you can find out by analytical chemical techniques to try and find out what the colours really were. I mean, there's water paintings of these, but they're all guesswork. So that's really been the basis of the TEXREC project. And Jennifer has been the key postdoc, Jennifer, or one of the key postdocs behind this. So it gives me a pleasure now to hand over to Jennifer, who's going to tell us all about this this wonderful project. Jennifer, it's all yours. So do you want to actually share your screen? We did practice this beforehand and it worked. I don't guarantee it's going to work now.
Right. Can you see that? Oh, hold on.
You see lots of papers that you're preparing.
Yeah, and it's not working. So much for the practice. Right. Is that okay? It's opening this time round.
Okay, so we can do a full screen on that.
Okay, can everyone see that?
Yeah, just two things before you start,
Can
Jennifer.
you see the presentation?
Yeah, can they make sure everyone has muted their microphones and preferably turned off their cameras. And if you've got to raise a question, the best way of doing this is through either raising a hand or using the chat facility, which is at the top of the Teams menu bar. And there'll be a chance to ask questions at the end anyway. And you can unmute at the end when it comes to the applause. That's how it seems to work. Okay, so all clear about that? Great.
Thank you so much for the introduction, Steve, and thank you everyone for joining. Steve, could you keep an eye on the chat, because I don't think I can see it when it's full screen, so let me know if I've missed a message.
Okay, no questions so far.
Good to know. Be worrying before I've even started. So I'm just going to give a little bit of background into the Oseberg first, what I did in relation to my PhD, and then on to the Oseberg and the textiles and the bulk of this presentation. So I'm just going to minimize that window. I don't know if you can see it. So as Steve said, this presentation is on the restoration of the Oseberg tapestries. And okay, seem to sorry, it froze there for a second. All right, hopefully that's the last issue. So this is a Viking ship burial. It was discovered in 1903, and the excavation started in 1904. So it was a burial of two wealthy women. They were laid to rest within the ship on a bed within a structure that was built around the bed. And it appears possibly being a feather mattress or something like that. There was a lot of feathers that were found along with a lot of the textiles. Around the edge of the chamber, there may have also been some hangings. However, the Oseberg tapestry refers to anything in the collection with any sort of motif on it. So they weren't necessarily all wall hangings. But the name so far hasn't changed. There's been a little bit debate whether or not they should change the name, but as a minute, it's just standing for all motifs rather than necessarily wall hangings. So a lot of these tapestries were actually found folded up near the bed, but because they're archaeological in nature, there's been over 1,000 years. They're in many fragments. So part of the TEXREC projects is trying to figure out which pieces actually link together in terms of the textiles. But one of the big things that interest the conservators at the time were the beautiful wooden artifacts. So these were given the priority at the time of the excavation, whereas the wool was seen as muddy, matted bits of unimportant wool, and it wasn't given quite the same regard as the wooden artifacts. So one person was interested, Frida Kraft, and she did some sketches, and she did some of the sketches in water. So she cleaned off a number of the artifacts to be able to conduct her sketches. And because they were also soaking in the water during the sketches, those have been cleaned significantly more. But I'll show you there's a mixture in the collection later. But the wooden artifacts were the priority, and the conservator at the time went all around Europe trying to find the best conservation method, and it appeared to be alum, which is a sulfated salt of aluminium or aluminium and potassium, and this forms a crystal inside the wood, and these crystals stop the wood from shrinking, cracking, warping as it dries, which allowed these pieces to be put back together. And some of the slates were in hundreds of pieces, and it allowed it to be reassembled. However, that alum produced sulfuric acid, which over time degraded the wood. So although they look in good condition from the outside, in the middle, it's like powder in the middle of some of the pieces. So the Saving Oseberg project, which is the project I was on for my PhD, was looking at ways of conserving these wooden, all the wooden artifacts that had been treated with alum. So the ship itself wasn't treated with alum, but it was a lot of wooden artifacts within the collection that was. And this sulfuric acid did a lot of damage. And one of the main method of conserving wood is generally polyethylene glycol, PEG. And you probably heard the Mary Rose was sprayed for about a decade. There was a big BBC documentary and things on it a good number of years ago now. But it was sprayed for a long time to allow this peg to soak into the wood. And that's the standard treatment for wooden artifact. And where there's no metals and acid, it does a very, very good job of preserving the wood. However, in cases where there is an acidic environment and when there is metal present, it can be more of an issue. And that's being found a little bit now with the Mary Rose and with the Vasa as well, which is another ship that was discovered. And because of that, there is concerns with the Oseberg artifacts about whether we should be looking at an alternative to PEG, because there's some metals, there's the acid problem, and there's also metals from the original artifacts and also from pins used in the restoration as well. So some of the artifacts are in much stronger condition and could support a water-soluble method, and then you'd wash away the alum and the acid, and then put something else in. So that could be PEG, or it could be something else that's water soluble. However, some of the artifacts are in such poor condition that if you try to wash away the alum, it would cause a collapse of some of the artifacts. So there also needs to be something that's not water-based, something organic soluble, something like alcohol or methylated spirits or something that would allow for material to be put back in. So what we were looking at was ethyl acetate, which is less toxic than some alternatives, but we found we needed to add a bit of toluene. So for the case we were working on, it'd have to be done in a fume hood. But it would allow the consolidant to be brought in without water, so the alum could stay in place. So we were looking at both aqueous and non-aqueous treatments. So this PhD involved looking at the polymers themselves, which these were polysaccharides, so large carbohydrate-based material that we were looking at. And we were assessing the material first on its own, and then we were assessing some artificially degraded wood and some archaeological wood that we did as test pieces to see how it would interact with the wood, whether it would fully penetrate the wood, and how effective it would be as a consolidant. So just to show you an example of the archaeological pieces of wood. This was part of the things to assess the wood. There was also various other tests. I'm not going to cover them all. But one of the things we also looked at is this addition of the silicon group here. And this is what allowed this to be soluble in organic solvents, which would allow the alum to stay in place. And having a look under the scanning electron microscopy with the energy dispersive spectroscopy, it allows to look at all the elements that are in there. So the thing we could search for specifically is the silicon. So we could see that it was around the edge of the cell wall. So the scanning electron microscopy gives you basically a really, really zoomed in image, a microscope image where you can see down to the individual cells within the wood. So we could see exactly how the polymer was interacting with the wood. So in this case, coating the edge of the cell's walls rather than completely filling the cells in most cases, which would allow the material to be retreated. Anyway, the wood is not the point of this presentation, but I want to show some of the history of where we come from with the initial research onto the wood. And I'm now going to be going on to the textiles and how I got involved, and this is where this feeds in. So the TEXREC project, so Steve has already given you a little bit of a background in it. So I mentioned the sort of puzzle program, trying to piece together the original tapestry. So there were multiple tapestries or multiple pieces of textiles with motifs. And they're now broken into fragments. So some of them came from the same, but there were multiples. Some of them came from other pieces. So it's like several puzzles have been mixed up together, and we're trying to piece together which bit went with which puzzle. So there's someone, David, in the project who's looking at computational techniques for the virtual reconstruction of these Oseberg textiles. And another part of the project is looking at interpreting the motifs and figuring out how they link to the Viking myth. And then there's the conservation strategies, was where I come in. So I had previously been working on these polysaccharides, these large carbohydrate-based material for the wood conservation. And we're looking at something very similar in terms for the wool, but more specifically for the wool rather than the wood now. So this is how I got involved in this project. And there's also a PhD student in Germany who's looking at some protein-based options as well, of a protein from mussels. So we're looking at different options for conservation. And the last bit of the project is doing the virtual reconstructions. So this will involve the dye analysis. So I'm doing a little bit of the dye analysis on the side of this project as well. So there's a lot of complications when it comes to conservation of woolen artifacts, archaeological artifacts. So generally speaking, the best method and what was being decided as the best method of conservation for the Oseberg artifacts currently is to have controlled humidity and temperature and to have a low oxygen environment to prolong the life of the textiles. However, there's a lot of stresses involved. Items in the museum are used for research. Sometimes it's necessary to be able to turn them over to look at the back. Also, in terms of exhibits, they need to be moved. So there's a fair amount of stress in terms of handling in a museum, and in the future, it could be deemed necessary to treat them with some sort of consolidant or to stitch it onto a backing material is the other option. But if stitching it onto a backing material isn't sufficient, then a consolidant is the only option left. So consolidation of textiles is generally very much a last resort option, where all the other alternatives are no longer possible. So because of that, there's been very little research on these methods and very little research, particularly more recently, going into more in-depth analysis of exactly how to compare it. So there's been a lot of trial and error in the past, and where they're finding out sort of 50 years later, oh, actually this didn't work and it's actually made it worse. So there's a big push now to do as much research as possible before going anywhere near the artifacts, so that the conservators have all the possible knowledge to base their decision on, rather than going by trial and error. So this is what this is to do with. It's getting that initial information for the conservators to make the best possible choice should a consultant be needed in the future. Then there'll be all the information ready to be able to choose which one to use. And there's also a wide variety of textiles in all sorts of conditions. So I mentioned that Sophie Kraft washed some of these to be able to sketch them. So we have the ones that are washed, where you have the individual single layer, or where the surface has been cleaned. And you also have the box like the one in the center, which they were just left and dried, matted all together. So there's quite a lot of variety in the collection, and each case is going to need special consideration on how that particular example should be conserved. So it's going to be a case-by-case basis for every single box in the collection. In terms of research, we started off with looking at dyes. So as you could see from some of the previous images, there's still some dye that's visible on some of the artifacts, generally mostly just red that's visible today, but I wouldn't want, and if this is used also elsewhere in conservation, I wouldn't want any consultant changing the color of the original artifacts. So the first thing to do was to obtain some modern machine woven wool, and the reason for starting with modern wool is there's less variability when it comes to comparing the treatments, so we know any difference isn't due to the inconsistency within the material, but it's actually due to the consolidant that's been applied. So we started with machine woven wool for that reason, and we looked at several different dyes. So Chaz, who's here today, helped me with the dyeing and showed me everything for this. So we started with madder and weld. So madder and weld are both plant dyes, so they're extracted from the plant, but they're both mordant dyes, so they require a metal salt first. So the wool is first mordanted, so this metal salt is applied and that attaches to the wool, and then it's then transferred into the dye bath, and the dye then attaches to the metal salt, and that attachment prevents the dye from getting washed away, so it's wash fast. And that's the case for mordant dyes. There are alternative types of dyes like vat dyes. So this blue here is woad dyed. So again, the dye compounds are extracted from the dye by heating it up in some water. And this is done in a high pH reducing environment, and that produces a water-soluble dye. You then insert the wool, and because it's water-soluble, it penetrates all through the wool. And as you pull the wool out, it oxidizes and forms an insoluble precipitate, which is blue, and that's what gives it that beautiful blue color. So you can see it coming out, going from sort of clear, not clear, but the original color of the wool, a bit of a green tinge to full-on blue. And again, because it's insoluble, it can't be washed out. The final type of dye that's been used in this project is lichen dye, that was kindly done for me by a master's student. She was doing a project, she's actually here today, on looking at the lichen dye and producing some material for studies and assessing, comparing methods of producing this lichen dye. So this lichen dye, or keel dye, is produced from lichen using stale urine, particularly from a man who's been to the pub the night before, is apparently, in some of the written methods, the best way of achieving this reliable dye. So the master student, Lena, she managed to find a volunteer who was going to the pub anyway to give her urine sample afterwards to be able to produce these beautiful bright purple lichen dyes, which are just absolutely amazing. It's so bright, the photo doesn't really do it justice. And so we have the samples, and that is a direct dyeing method where the dye attaches directly onto the wool. So that allows us to cover all the different types of dyes. So it reduces the chance that we miss some sort of interaction between the consolidant and the dye by covering all types of dye. And then we were looking at how best to apply the consolidant. So we decided to go with a spray application. We did consider immersion or sponging the dye, the consolidant on, but it is a bit of a problem. They each have their own positives and negatives, but at the end of the day, if it came to an actual artifact, the most likely way to treat it would be to spray it, or use a misting chamber and have ideally a vacuum underneath to draw through the mist through the fiber. Unfortunately, we don't have that instrument available to us, so we decided to go for the next best, which is looking at the spray option, and we needed a spray that would be as even as possible. So again, we are comparing like to like in terms of the amount of consolidation. If the evens on spraying, then it'd be comparing the difference in the amount of spray rather than the difference in the consolidant. So because of that, we went for a paint spray gun instead of some of the other spray options. But I have to say the pump sort of oil spray for food actually worked quite well as well. The spray gun, unfortunately, you could see the wool move a little bit under the pressure from the spray gun. But like I said, for this first initial test, we wanted something that would give as consistent results as possible. So that was a sort of middle ground for that. And And then we were looking, so now it comes to the actual treatment. We were spraying 5% because it would allow it to pass through the spray gun easily and not be too thick. And we were doing 4 applications. So I looked at hydroxypropyl cellulose, which is used in conservation, methyl cellulose, which is also used in conservation, and phenori. So phenori is, again, a polysaccharide, but this time from seaweed, whereas the others are sort of modified from cellulose naturally found in wood. And the phenori method has been used a lot in Japan, particularly for cellulose-based material, particularly for paper, and some other textiles as well, but largely paper. But there has been some use of it in silks as well, which is also protein-based. So because of that, it could have the application, it could be suitable for wool as well. So we wanted to compare those, and because of the work we've previously done on the Saving Oseberg project, and that looked largely at chitosan, which is from crab shells. So because of that, we were looking at these water-soluble modified chitosans, which we'd previously ruled out for the Saving Oseberg project because we were predominantly looking for something that was organic soluble, and this didn't fit the bill. But for the wool, we were looking for something water soluble, so we decided to have a go with these as well. And the chitosan has a similar structure to cellulose that's found in wood, but it has an amine group, and that amine group can attach to metal ions, so it could prevent some problems coming from the metal ions and the migration of the metal ions. And the amine group, in terms for the Saving Oseberg project, it could also help to reduce some of the acidity as well. And then lastly, we were also looking at microfibrillated cellulose. There's been some promising research come out the past few years using this for paper conservation, so we decided to also include that in our research. And then we were comparing the results of those consultants to untreated wool and to water control to double check the actual application process of the consultants wasn't also having an impact. And then, so we had to find ways of assessing the results. So we're looking at weight and dimensional changes. So the weight changes is more to assess the variability within the spraying method and between the different applications, and looking at the dimensional change of the material as well. Then looking at the visibility of the fiber under SEM to see, can we still identify the fiber under the SEM, or would the consolidant interfere with that? Determine the presence of the consolidant using infrared spectroscopy, and then looking at the stiffness of the material. How much stiffness does it add? Does it make it noticeably stiffer? And would that make the handling easier? And then we're looking at water absorption. How does it retain humidity? Looking at the color change, the reversibility, and also artificial aging. So like I said, there's been some issues in the past with consolidants that turned out not to be positive, or no consolidant will also last forever. So sooner or later it might need to be replaced. Or also for analytical testing, you might want to remove the consolidant. So that reversibility aspect is important, to be able to remove the old consolidant to be able to apply a new consolidant in the future. And then also looked at artificial aging to see how well would it age in a museum. Because we want something that would last as long as possible, because every time you try to remove a consolidant, you risk damaging the artifact. So you want to, if you want it reversible, but you also don't want to have to remove it. It is, again, when there's no alternative, you remove it. So you do want it to last as long as possible. So I'm just going to go through the scanning electron microscopy. So this is down to, you can see here is the individual fibers from the wool. And these marks along it are the scales, and that's indicative of a protein hair fiber. So this shows we're still able to see that. So we're still able to identify it as wool. Whereas with plant-based fibers, like linen, you wouldn't see these markings. So it's still possible to determine what type of material the artifacts were made of, even after the application of these consolidants. The only one where it's really difficult to still assess it is with the microfibrillated cellulose. With the unpurified phenori, it's still visible, but it's covered up by some of these impurities. But we did compare not with a spraying method, but with an immersion method, which was a different part of the project. We did look at a phenori extract that we purified ourselves, and where you can see the fibers much more clearly, and also looked at a commercially available purified phenori. And you can see those impurities are no longer there, and you can still see all the scales on the wool. So these images go down to about 50 micrometers, and they're incredibly small. And like I said, looking at the individual fibers to be able to assess what the material is made of. We also wanted to know whether or not we could see the polymer on the wool to be able to identify it. So if any records are lost in the future, to know that it is possible to still identify what consolidant was applied, and also to know that we did successfully apply it. So this orange line is the untreated wool, and the red one is the polymer. So with this blue one, that is where you can see that it was the coating was visible on the polymer, and you can see it matches with the polymer. So this peak there is not present in the untreated wool, but is once it's treated. So, and it matches the polymer. So this is only possible because the infrared microscopy that we have also has microscope attachment. So we're able to look specifically at the coating, because otherwise there was such a strong signal from the wool that we're struggling to be able to identify the consolidant. But with the microscope as well, where we can narrow down exactly where to look, where we can see there's some consolidant on the surface, that allows for the identification of the polymer. So you'll be pleased to know I haven't got 20 images of these. I've just got this one as an example. And I will move on. So there's the next thing we're looking at is assessing the stiffness. So what's done in engineering, they use a cantilever to be able to assess the rigidity of materials. So unfortunately, we did not have one available to us. So I decided to construct one out of PDMA, just these plastic sheets that the museum had. So I asked someone in exhibitions if I could take some of the plastic sheets that they were using and cut them up. So they kindly gave me some, and I cut it up and constructed this unit with a 45-degree angle that you have on a cantilever. Because it's self-constructed, can't compare it to literature results, but we can compare it within our own tests. So everything was done on the same unit to be able to compare within our project. And we compared everything to the untreated and the water control. And this could be used in any conservation lab. It wouldn't be restricted to engineering or having to buy the unit. And what happens is you push the material along here, and then it drops, and you measure that drop. So if it's very, very flexible, it will immediately, the second you push it off the edge, it will drop onto that 45-degree angle. The stiffer material, you'll push out further before it starts to bend and drop. So that's a way of assessing the stiffness. So these are the results from that. So this flexural rigidity also takes into account the weight and the dimensional area as well as that bending length. So it takes all of that into account so that you're not having any bias from the exact size of piece that you cut. So if you look at the undyed, the lowest numbers represents the most flexible material. So these were very, very flexible. And when you add Kusal-G, you've got a slight increase in stiffness, but the Finori by far resulted in the biggest increase in stiffness. Now, this stiffness could be very, very beneficial when items are to be laid flat, because you'll be able to turn them over without bits that are loose bending under their own weight and potentially breaking because of the brittle nature. So this could allow for more handling, and this is important for exhibitions and also for research purposes. Now, there could be debate about how much stiffness you want. Feeling all these wool, they were still very, very flexible, despite some increase in stiffness, but all the material was still flexible. Now, if you're looking to drape the material to show how a material was originally used, in the cases where it's in good enough condition to be draping, you wouldn't be applying a consolidant in the first place. You'd be stitching it onto a backing material. So this is only really for cases where you'd be laying it flat anyway, because it is the most fragile of pieces. So that increase of stiffness is positive in those cases. But you can control exactly how stiff it is by how much consolidant you apply. The more consolidant you apply, the stiffer the material will be. But also which consolidant apply would have a difference as well. So if you're wanting to have a little less rigidity, you could go for the Kusal-G instead of the Finori seaweed. Then we also then looked at the color change. So the microfibrillated cellulose was a complete disaster. It produced a white layer over the surface of material. So in cases where it was for paper, that wasn't so much of an issue that was opaque. But in terms of the woolen textiles, you want it to be completely see-through to be able to see through it. So that was very much a problem, and for all the dyes. So when we don't include that, we can then zoom in a little bit more on the rest of these. And then we could see, now there's a lot of variability in here. So for anyone who's doing dyeing, I'll probably be mocked a bit for this, but I wanted enough material for the whole project, and I wanted it all to be in the same dye bath, so it would all be the same color. Unfortunately, our pots weren't quite as big. So because of that, we've got quite a lot of variability within the same piece of wool. So that is what accounts for the large variability here. Everything was compared to the untreated. After I'd done these measurements, I did find a better way of quickly or slightly more quickly measuring the color changes, which would have been more suitable for doing before and after. But because of the time involved, compared everything to the untreated, and afterwards it was too late to go back. But what is clear, despite this huge amount of variability within the samples, this lichen dye, there was less variability within the lichen dye. And this, you could clearly see a change compared to all the other lichen treatments. So the hydroxypropyl chitosan produced a large color change. So that is problematic for the lichen dyes. And for the carboxymethyl chitosan, it was the yellow that produced a large color change. So that is an issue. Like I said, if I was to repeat this, there's always going to be a bit of variability with the dyeing. Even if it was a very small piece, there would still be variety across the material. So to repeat this work, we'd look more precisely at the exact same points. And that will be able to zoom in on some of the more detail, but it very much does highlight this is for the well that is well out with what's seen in all the other ones. So this carboxymethyl chitosan is definitely impacting the color, and it was making the weld a lot brighter, whereas the color changes from the other treatments weren't terribly visible. But there was a big difference with the weld. It became a lot brighter after treatment. So you could argue whether that's positive or negative, but at the end of the day, it is a change to the color of the original material. So that would have to be taken under consideration. So in terms of the reversibility, I was looking at seeing whether the treatments were reversible or not, and wanted to see if you could wash out the consolidant effectively. So we're looking at the water control, and there was a big so we looked at washing out. We're comparing the weights before it was treated to after we washed out the material to bring it back to whether it came back to the original weight before it was treated or not. So you can see there's some small variability just within the water control, but it's pretty much gone back down to zero. The couche algae has also gone back to original weight and lost a little bit more, and that could be because some of the fibers that were loose around the edge possibly came off during the treatment or during the washing or during some of the measurements as well. And what can be seen, they're mostly reversible, particularly the phenori is very similar to the water control. What's very interesting is for the carboxymethyl chitosan, the one that we saw the color change with the weld, particularly for the weld, that weight hasn't gone back down to what was before it was treated. So for some reason, it seems to be holding on a bit more to when the weld dye is present than with any of the other dyes, which warrants further investigation. It's kind of interesting, but not particularly relevant. The point is that it is retaining that and it's not fully reversible. The microfibrillated cellulose is not reversible, but I didn't expect it to, because in that case, it's not dissolved in the material. It's particles. So it's no surprise that it then doesn't wash back out. And again, this could be problematic. In terms of artificial aging, we tried to replicate the light exposure for the sensitive organic artifacts. So that would be 1,500 microwatts per meter squared. And we wanted to see for sort of 8 hours exposure a day for 100 years, 110 years, whether or not we would see a color change. So we used a light, very strong light for 8 hours to sort of replicate that. And what we were looking at is we couldn't see any color change that was visible to the naked eye. But with the spectrophotometer, which measures that color change, you could see more subtle changes. And you could see the methyl cellulose was the one that was impacted most by that UV light. Seemed to be the least stable of the lot. But saying that, all the treatments were acceptable because there wasn't any visible color change even after all that exposure of UV light. So the microfibrillated cellulose caused some yellowing and some darkening. And the phenori caused a little bit of yellowing, where the couche algae was a little bit on both sides. So in conclusion, the microfibrillated cellulose produced a white film, so it rules it out for a consolidant for wool. Hydroxypropyl chitosan produced a color change with the lichen dyes, which again can be a problem. So if there's any sort of suspicion of pH sensitive dyes, it can't be used. Same with the carboxymethyl chitosan, where there's presence of weld, again, this warrants a bit of further investigation, but caution should be taken if choosing to use this as a consolidant. The phenori seem to be the most favorable in terms of color change and increasing that stiffness to make it easier to handle. But but there is a variety of different ones, and the CUSAL-G was also gave very positive results. So if it was desirable to have material that was a little less stiff, that could also be an option. So we've provided the conservators with hopefully a lot more information to make their choice on as to what stiffness they want, what polymer to use. So the next stage will be looking at temperature and humidity aging, looking at tensile tests on treated and untreated wool fibers, and then looking at treatment of degraded wool as mock-ups for archaeological wool. And then that will be to understand a bit more about the consultant ability as well. The next stage, so the next part of this project was looking at the multi-analytical approach for looking for the dye analysis. So we're moving on from sort of consolidation side of the project onto the dye analysis that I did. So these are some of the reconstructions of the tapestries. So you can see there's variability in the choice of colors for the tapestries. And all that's really visible today is the red that you can see, particularly on the horse. But this red is still visible, but all the others are sort of varying shades of beige and brown. So these colors were very much guesswork in some of the reconstructions other than the red. So the aim is to determine chemically what are these dyes that were used and what color would they have produced to try and be able to do some more accurate reconstructions. So I wanted to zoom in on this a little bit more so you can see these figures and the different colors. The example that we're using to start with is this TEXREC 7. So this particular, we took two samples, one from this sort of reddish brown thread here, and one from a more brown color to look at the two colors that were within this border. And we started with SEM-EDS. So that looks at the elemental components of the material, looking at carbon, oxygen, nitrogen, and sulfur that you find in wool. So we've identified all the components of the protein in wool. But we're also able to see iron and aluminium, which can be used as mordants. And in the case of historical textiles, that would help you to identify the mordant. The problem is with the archaeological material, because it's been buried, some metal ions from the soil could go into the woolen objects. Therefore, these presence of these metal ions doesn't need to be taken with a pinch of salt. You have to consider, you know, has it migrated from the soil? Could it be from nearby corroded artifacts as well? These two samples are close by. They're not from two completely different artifacts on two different areas of the site. So it's more likely it's from the mordanting, but you can't rule out whether there are other factors at play. Another analytical method that can be used and is commonly used for identification of dyes is LCMS. So that's liquid chromatography mass spectroscopy. And that allows you to separate out the compounds through a column. So the compounds interact with the stationary phase at the edge of the column while you pass a solution through it, the mobile phase. And it's that interaction between the phase that's stationary and the mobile phase that separates out all the different compounds. And the mass spectrometer then identifies the mass of each compound that comes out. So knowing how long it's taken to come out and knowing the mass allows you, particularly if you've got standards, to identify what the compounds are. In some cases, you can also have MSMS, and that's where you identify the mass of the compound, but you then also break down the compound, which gives you a sort of fingerprint of the different masses that make up fragments of that compound. And that gives you sort of like a fingerprint analysis of what the compound is. So you can then match it to a database. if you create a database of all the different dyes out there, you can then match it. So you have a sort of third confirmation of whether or not it is your dye. But this requires larger sample size than what would be really suitable for some of the ones with these figure motifs. You couldn't take samples that big from these figures. So that's why I looked at possible alternatives, and I found out through Nottingham we had access to OrbiSims. So TOF-SIMS is time of flight secondary ion mass spectroscopy. So that sends an ion beam down, produces a secondary ion that goes to mass spec. So with a time of flight mass spectrometer, that's a type of mass spectrometer. This OrbiSims is an Orbitrap mass spectrometer, which is a slightly different, more newer type that gives very, very high resolution. And it can give you more precision. So I'm going to come back to that later. But what you do is you send this primary ion beam down, you get these secondary ion beams, secondary ions that come up, and that goes to mass spectrometer, and you identify the mass of every ion that comes up. So it gives you a fingerprint of everything that's in that material that you're analyzing. And with the OrbiSims, you can also do a depth analysis. So if there is a coating of consolidant over the material, you could theoretically go through that layer onto the actual fibers to identify the dyes. And you can also do this fragmentation pattern of each individual compound, like with the LCMS, to give you, again, this fragmentation pattern that you can search through databases. Unfortunately, in practice, the dyes prove to have too low a signal to be able to look at this fragmentation pattern, but we still can identify the mass of the compounds. Unfortunately, there are a number of compounds that can have the same mass. So what we do is we search for dyes, and we look to whether or not they're present or absent. And if they're present, then there's a high possibility that dye was used, particularly if we can see it wasn't we don't see it in the untreated dye and not in every single archaeological material to confirm it's not a breakdown product of something else. But in terms of these flavonoids, that causes us a particular issue. So there are some plants that have a combination of these, and it's the different combination that allows you to identify specifically what plant. So that's where OrbiSims struggle. So we can tell some of them, and we can tell there's a flavonoid that seems to be present, but we can't identify exactly what one. These ones, for example, all have the same mass. So we can tell that it was yellow, but can't identify specifically the plant. So ideally, what you do is take a sample from where you can take a sample specifically exactly where you want on the tunic, for example, and you can identify the color of the tunic. And you can then take a sample from one of the boxes in worse condition and narrow down exactly what plant was used. So doing a combination where you can get the suitable size. So I just want to quickly show you the difference between TOF-SIMS, which has been used before for analysis of textiles, and OrbiSims. So like I said, the only difference is really it's a newer mass spectrometer. It's the same as looking at the surface and looking at all masses that are present in the material. But because this is a slightly more modern instrument, it gives you much higher resolution than this instrument. So, but it also has a slightly different mass range. So there are situations where you'd use the TOF-SIMS and situations where you'd use the OrbiSims. But for us, this OrbiSims is particularly beneficial because it allows us to look at the complexes of dyes and the mordants together, rather than just one or the other. Whereas LCMS, the first type of mass spectroscopy I talked about, where it separates the compounds, that can't look at the mordants, and the scanning electron microscopy with energy dispersive spectroscopy identifies all the elements, so can identify the mordant, but it can't identify the dye. So the Orbisims can do both, and it uses much, much smaller samples, which is why it's particularly interesting for this project. So we then search for specifically for the dye compounds that you find in woad. And this indigotine is what's present in woad. It's also what's present in the indigo dye that's from India, but woad is one that's more generally accessible in Europe. And what we found is we didn't find that mass present on the undyed wool, which reassures us that when we do so, it is because it's from the woad. And we were also able to look at breakdown products and determine whether or not these masses were present. And what we found is in that second sample that was taken, the one that was more brown, we did find all these different peaks. So because we're searching for multiple compounds, we have more assurance that is indeed from the woad rather than just looking at one compound. We also looked at madder. So madder was the plant that gave us the red color, and alizarin, purpurin are compounds that you find within this madder. The problem we had is we found this even in the undyed material and across all samples. So there's something else present in the wool that has a similar chemical formula to the compound of madder. But what we did find was with the complex, with the aluminium, that was not at all present in the undyed wool and was only present in the madder dyed wool. And it was also present in both samples from sample 1 and sample 2. So it shows madder was used, and it was used specifically with aluminium. And aluminium and madder gives quite a bright red color. So that brown would have been a red color. And this one, which was brown, was treated with madder and with wood, which gives a purple or black, depending exactly the ratio of madder and wood that's used. So just to show a little example, I've chosen to color this in a sort of dark purple, almost black, as an example. We don't know the exact shade of purple. That we can't determine. What we can determine is what chemical compounds were found and what we know what plants contain those compounds. So it does seem to have been madder and woad, which, yes, could have given a purple or black. But I just wanted to show you, just with this border sample, that we can reconstruct what the original colors of this border would be. And if we had somewhere else where there was a small part of the border visible, we could test again and see whether we're seeing the same colors to identify whether it came from the same original tapestry or not. And hopefully we can also then go on to test some of these other parts. We can look at the individual figures to look at the colors of the tunics or the spears or the horses and determine and reconstruct the color of the whole scene. And with boxes like these, we can't identify the exact location, but what we can do is use these samples for LCMS, where we can take much larger samples and narrow it down to the specific plant. Particularly for the yellows, which we can determine it's flavonoid, it's yellow, but we can't determine which one. We can look at these boxes and then hopefully use those to determine exactly what plants were used within the collection. So I just wanted to say thank you to everyone for listening. I hope you find it interesting. I certainly did. I really loved working on this project. I think it has a lot of potential to look at all the different colours. And I want to thank Hartmut Kuzel, who is the PI on the project, and Steve Harding, who is leading it from Nottingham. Anna, who's the expert in the Orbisims at Nottingham, and Hannah, who's the conservator, and Chaz Jones for helping me with all the dyeing, helped me so much, and me being quite stubborn about how much material I wanted to dye. And Lena as well for giving me some samples with the lichen dye as well. It was really beneficial to have that in addition to ones I'd already dyed with Chaz. And to everyone at the MCMH and Colin and Paula, who are also in the project looking at other aspects of dye analysis. So I will just end it there. And does anyone have any questions?
So, wait for the, hang on, Jennifer, wait for the applause. Everyone unmute. Unmute. And then away we go, folks. What? You can use there's actually a clap function on the scene. Right.
Thank you.
So thank you, Jeff, on behalf of all of us. That was Carl's found it. That was fabulous, and it just shows you the tremendous amount of science that's involved behind all this. Not just the conservation work, but in identifying not just the color of the dyes, but the plants. And as Sudha said, you know, that will enable us to find out not only where the sheep came from in terms of the protein and the analysis on the wool fiber, but from the dye analysis and plants to get an idea where the dyes came from. Exactly that picture of that washing line with those different types of dye material. Is that Chaz's backyard? That's Chaz's. I recognise that. Okay, right. We've got time for questions now. What's the best way of doing this? We can either check the chat.
Oh, yes.
Or we can just unmute. Claire just said, great, really interesting, great combination of archaeology and chemistry. Thank you. Yeah, that's exactly what it was. It was a great combination. Yeah. Chas is gone. Oh, Chas had to go away. Yes. Yeah. Right. That's a shame. Anyway, he says it was great. So, any questions? Anyone got any questions for Jennifer on this? Well, obvious one. Sorry, Jennifer. Is how long it's going to take before we get a definitive answer.
As long as it takes to find out about Brunenburg.
Because I don't think scientists take a long time. Conservators are very careful and cautious. And,
yes. So
we
we have actually, well, we have actually got permission to do some more sampling. So hopefully some of the Orbisims analysis will continue. So it may not be me doing it, but there's going to be some continuation of that by the sounds of things.
Yeah.
And some samples for the LCMS analysis as well to more specifically look at some of the plant that could have been used as well where Orbisims might struggle.
Claire's just typing something, actually. Yep. Claire, you can unmute your microphone if you like. If you can.
Yes. Hi, thank you so much. It's really interesting. I was wondering if you could use those techniques on different sorts of fibres as well, like ancient silks and cottons or something like that. Are they applicable?
Yes. So I think the TOF-SIMS method, which is the other type of mass spectrometer that's been used, I think that's been used before, and I think that might have been on silks, if I remember right. So it can be used on different materials, but the Orbisims is quite new using this newer MS. So I don't think it's been done before, but there's no reason why it wouldn't be possible.
Yeah, but some of the Oseberg stuff is silk, isn't it?
Silk,
it's not all.
Yeah, particularly to do with origins of the silk. Origins of the dyes you were talking about earlier, Steve, to know exactly where the dyes were from. I'd be particularly interested for some of the silks as well for that, to be able to determine where the silks had been traded from.
Yeah.
Yeah.
Go on, Peter.
Hello, thank you very much. By the way, my background is in organic chemistry and X-ray crystallography, but that was 60 years ago. But the question was, there's been some recent analysis of the Bayeux Tapestry.
Oh, yeah.
You saw a program, we saw it on BBC4 about in November, where I
Okay,
think
what's that? Chaz had mentioned there had been some analysis.
yeah, it was. I think they were using infrared spectroscopy to look at the dye
Okay.
compositions, and this was a PhD thesis of a lady who was at the Sorbonne University. I was wondering whether you'd come across any of that work and what you thought whether infrared spectroscopy.
I haven't. We did go to the synchrotron and try to do some infrared analysis on a standard FTIR. I don't think it would be possible to see it because you get such a strong signal from the wool and there's so little dye in comparison that the signal's not strong enough, which is why we went to the synchrotron to do some of the analysis. The results aren't back yet. So we're still waiting to do some of the data processing from that. So I don't know the results, unfortunately. But I think we could still see a very, very strong signal from the wool. But I'd be very, very interested to read that Bayeux one and pass it back on to Hartmut. He'd be fascinated.
Look up Clarisse Chauvin from the Sorbonne, and it was her PhD thesis.
Okay.
I think that's the one that Chaz spotted, Jennifer. Remember that one we tried to
the same one, yeah.
Yeah, track down. But we tried to get in contact with her, but without much success, actually. Maybe because of our bad French.
Chaz or we have had no success, but we downloaded her 230-page thesis from the internet.
Yeah.
We're still fighting through the French.
Yes. Well,
job is mostly chemistry.
Okay, my grandchildren decipher that. They're French. Maybe not at the age of 5, though. It's a bit ambitious. Great. Does that answer your question, Claire? Yeah.
Yes, that's great.
Okay, great. Anybody else want to make any points to Jennifer while we've got her? Or about the Oseberg ship itself and the artifacts, which is another story, really. I mean, I guess the tapestries are quite easy. That's the right word, and not easy at all, but easier compared to the problems with the Oseberg burial artifacts.
Yeah, we don't have the same problem with the alum or penetrating into the wool. This is a treatment that's going onto the surface of the wool. So it does simplify matters a little bit.
Yeah, but some of those boxes of material are in very, very bad condition.
Yeah.
How the Trondheim guys have managed to reconstruct or
to be fair, they are having trouble. They've done some very good job with some of the modern examples. So they cut up some modern bits of fabric, and it did an amazing job of doing that. But a lot of the work was from the differences in the color, which in the original Oseberg tapestries are faded, so they are struggling a lot more with the pattern matching from that. So they're looking at slightly different approaches and adding in multiple factors to be able to try and do it a bit more like the human eye does it. So it'll be interesting where that goes next as well.
Sue's got a jigsaws, maybe she might be able to
join us.
Anyone got a jigsaws? Yeah, it really is. Of course, this is Norway's national treasure we're dealing with here. I mean, Stan knows about this. How much they value the Oseberg ship more than anything else, really, isn't it? And all the artifacts and the textiles, because the text, all the imagery there, that just contains all sorts of information about what life might have been like during the the Viking age. It's not just about how they made their textiles, but the imagery which came with this has stories to tell, which is being interpreted like the picture with the Viking person with horns. So, you know, it's not all just down to Wagner and the Victorians. There's someone there with so you know what's he doing and or what's she doing and things. So these are absolutely fascinating pieces, and which is why it's worth all the effort in trying to preserve them and understand what they are. Yeah.
How old are these tapestries, by the way? When would they have been constructed?
So we don't know. The burial, the frame surrounding the bed that was made for the burial chamber, that was 834 AD. That's been dendrochronologically dated to. But the textiles, I mean, they're obviously not newer than that, but they could potentially be older if they've been passed down for a few generations before being placed in the burial. So there is talk of possibly trying to carbon date some of that, but it's not been done yet.
Okay, thank you.
That should be relatively easy, actually, from because the wood, the wool fiber, keratin and things looks in reasonably good condition.
Yes. The problem is, so there are some pieces in the collection that have been conserved in the past. There was quite a lot of conservation done. Was it around the 40s and 50s? So that could interfere with the radiocarbon dating. It will be dependent on how easily we can wash off this old consolidants or find a piece that definitely hasn't been consolidated.
Right.
So it will, I mean, it should be possible, but it'll be a matter of finding the right piece to analyze that's not contaminated.
Yeah. Great. Any other questions for Jennifer? I mean, you've done a really great job, I think. I mean, the whole team is a great team, actually, a mix of conservatives and scientists working together, which is not always easy, trying to understand each other's language and problems, but it seems to work well. And let's hope that it gets, because it's been supported by the Norwegian Funding Council. Let's hope that's
I hope it continues.
Funded, it's I think it's great stuff. So once again, can we thank Jennifer for this excellent presentation? It has been recorded, so we'll have it on YouTube, so the link. And once again, thank you.
Thank you.
Thank
Thank you very much.
Hope you enjoyed it.
Bye bye.
Okay,
Bye-bye.
and the next presentation series will be by, I think, Paul Cabell in July, is it? Who's a etymologist, that's the place name expert, and yeah, there'll be a bit of Brunenbury in there as well, I suppose.
No, where it says leave.
Okay, right, see you folks.
Bye bye.
Bye. Bye.
Bye.
Stay safe.
Yeah. Be cool.
Bye.
Okay, Jennifer's gone. She wasn't supposed to do that.
But he's got her,
Michael Lynn is still there, yeah.
she's got her nieces and nephews to bother about.
Yeah, Dale, that's the Norwegian guy, I think. Okay, guys, we'll say goodbye. Thanks very much for joining us. And any feedback you have will be much appreciated. I'm going to stop the recording now. Okay. Stop recording.